Hi everyone, Mrs. Below here. In this video, I'm going to be going over the cell division notes, slides 36 through 41, over cell differentiation and stem cells. So we of course have been talking about the process of cell division, going from 1 to 2 cells, and that includes going through the cell cycle, interphase, which is broken down into G1S, G2, mitosis, which we break down into PMAT, prophase, metaphase, anaphase, telophase, and then cytokinesis. And that's to make more cells in the body, in somatic cells. And those cells are diploid. And in terms of getting a little bit more specific with this, we can also talk about cell differentiation, which is when these cells are dividing, they are becoming specific types of cells. So cell differentiation is the process by which an unspecialized cell, so a cell that's kind of like a blank slate, develops into a specialized cell. By specialized, I mean it's a specific type and therefore will have a specific job. So like skin cells, for example, it's called they're squamous, so they're flat, they have this flat structure, and it's so they can layer on top of each other to create a protective barrier between the inside of your body and the outside of your body. So that's an example of a specialized cell. Remember, cells have a specific shape, which relates to their job or function. So how do we tell what a cell will become? How do we tell it to do that? Well, this is done by turning on or off specific genes. So in your DNA, right, you have all 46 chromosomes, and you have all of that DNA in every cell in your somatic cells in your body. However, you're not using every single gene that's in your DNA. It depends on the type of cell. Certain cells will turn on and off specific genes. For example, skin cells contain keratin, which is a protein that's in your hair, skin, and nails. And that is a gene that would be turned on in a skin cell's DNA to make the protein keratin. But something like, a bone cell wouldn't necessarily have to turn on that gene. It's going to turn on different genes to make whatever those bone cells need. And what determines what a cell will become is based on its location in the developing embryo. So I have a video to kind of illustrate that. So this is a National Geographic video, and it's a salamander developing from a single cell into a full-grown salamander. And this illustrates this process very well. So here's a salamander. It is a single cell. So egg and sperm combine together, and we get a zygote, beginning stages of life. And this cell, you can already see it's going to go into mitosis. It's going to go into cell division. So that one cell you can see is going to become 2 cells. And then those 2 cells become 4 cells, so on and so forth. And you can see how those cells keep dividing. And then we're going to get kind of this ball of cells. I'm going to fast forward a little bit. So now you can see there's a lot more cells, right, that came from that single cell because they're going to keep dividing. Now, these cells are going to start to turn, they each have their own DNA, right, all 46 chromosomes. And they're going to start to turn on and off specific genes in order to become a specific type of cell. Let me fast forward a little bit. Here we go. So there are the cells that are kind of closer to the outside of the embryo. Those are becoming skin cells. The ones on the inside are becoming muscle cells, bone cells, intestinal cells. And that's done by turning on or off specific genes in the DNA, and that depends where those cells are located in the developing embryo. And eventually, we'll get this little tadpole-looking guy, which eventually will become a full-grown salamander. Let me pause the video. So that is kind of where cell division comes in and cell differentiation comes in. Cells divide, make more cells, but then they become a specific type of cell by turning on or off a specific gene, and that depends where it's located in the developing embryo. All right, now stem cells, these are really interesting area of research, really interesting type of cells. And the reason for that is because they can do a couple cool things. So one is they can divide and renew themselves for long periods of time. They're, to some degree, you could say almost like immortal cells because their telomeres don't always get shorter. Not that those telomeres never get shorter in stem cells, they just, like it's a little bit slower of a process than normal cells. So they're kind of, I guess you could call them immortal cells in comparison to normal body cells. They just don't age as fast as like other cells in the body. They can also remain in an undifferentiated form. So meaning they're basically this blank slate, they're an unspecialized cell, which is nice because then that cell can divide and either make more stem cells, or it can divide and one of those stem cells can stay a stem cell and the other one can become a specialized cell. And you can, depending on the type of stem cell, it can develop into a variety of specialized cells. Okay. So then that's how you can basically renew some of the cells in your body. And there's different classes of stem cells, right? This is like just a generic example that have differences in terms of their potential to develop into different types of cells. So for example, if you have like cells that are coming from like an embryo or a fertilized egg, those usually have a lot more kind of wiggle room to play with in terms of like being able to manipulate them to become a specific type of cell. In research, right, we can extract stem cells from different areas and we can use them and make them become a specific type of cell for whatever like a patient might need. And I'm going to summarize the differences between these on the next slide here. So there's kind of two areas that we can get stem cells from. One is adult stem cells. So that would be from like, if I'm talking about this from a medical perspective, this would be from the patient themselves. And, you know, as an adult, as a human being, you have stem cells and potentially you could go in and get those stem cells in the body and use them, manipulate them to become a specific type of cell that's maybe whatever the patient needs. Maybe there's a certain organ or a certain type of cells in the body that are affected. And maybe you can get those stem cells and make them become a specific type of cell that's a healthy version for the patient. The nice thing about this is there's a very low risk of transplant rejection because the cells are coming from the patient themselves. So the DNA is the same. So very low risk of rejection. It's kind of like when you get an organ transplant. If you get it from another person, the body could reject it because it doesn't recognize it as its own. But if you're getting the stem cells from the patient themselves, then the body most likely won't reject it. This also avoids some of the ethical issues that are associated with embryonic stem cells. I'll get to that in a second. However, in practice, there's very few in number. These are very difficult to work with. They're very difficult to grow and isolate. And again, there could be some abnormalities in those cells. So pros and cons, advantages and disadvantages with adult stem cells. Embryonic stem cells, so yes, this would come from an embryo. This would come from something that has the potential to become, you know, a human being, a baby, right? Which can definitely be, you know, an area of ethical concern because, you know, I mean, I guess it depends where it comes from, you know, but it's coming from an embryo. Which usually in the process of getting those cells, it would destroy the embryo in the process. So there is some ethical issues associated with that. However, these are very easy to work with because they can become almost any type of cell. They're very easy to work with, very easy to grow. However, there is a higher risk of rejection because if you're getting those cells from an embryo and putting them into a patient, it's kind of the same idea as an organ transplant. They could be rejected, and there could be some other issues associated with it. So again, kind of pros and cons to both types of stem cells. Also, stem cells could be used in cloning. So somatic cell nuclear transfer. Let me kind of explain this process. So yes, this is used to clone Dolly the sheep was one of the first cloned animals in 1996. So here's kind of how this process works. SCNT, somatic cell nuclear transfer. So you have a Dolly, you have the original kind of sheep, okay? And so this one right here, and you take their DNA, you can see their DNA here, and you put it into a donor egg whose DNA has been removed. Remember, an egg only has half of the DNA, right? It's haploid, and you can see here it's been removed. So basically you have like this empty egg. Like it's still the egg, it's still the cell, but it doesn't have the DNA from the donor anymore. And then you can take the full diploid count, all of those chromosomes, and put them into this empty egg cell. Well, now instead of doing like egg and sperm combined together, you're taking all of that DNA from the donor or from the thing you're trying to clone, and you're putting it in that egg cell. And then you stimulate that to grow. You put it into a surrogate. So then eventually this would become identical to the thing you were trying to clone. Obviously, they're going to be different in ages, but they would be identical to each other, and that would be cloning. Now, in terms of how this could be potentially used for humans, again, this all sounds good in theory. In practice, it's a lot harder. But in theory, this is how we can kind of use that idea of cloning to help humans in a medical sense. So what we could do is we could take a human egg cell from a female and we extract the DNA. So then you have like this empty cell. And then you could take the DNA from the patient and you can put their DNA into that empty cell. And instead of using it to like obviously clone a human, which that's a whole nother ethical issue, we could just use that, we could stimulate it to harvest those stem cells and then use those stem cells to make whatever cells that person might need. Maybe we can use it to grow an organ or new types of cells that the patient needs. And that avoids the ethical issues of the embryo, because we're just using an egg cell that hasn't been fertilized, that didn't have the potential to become a baby. And we have a low risk of rejection because the DNA that's inside of this is coming from the patient themselves. So really good. In theory, actually doing this is a whole nother feat. But that's the idea behind how we can use cloning, somatic cell nuclear transfer in a medical sense to help